Okay, we are talking about reaction mechanisms, and as we understand it, it's essentially a proposal. So one of the things that we have to be able to do is to confirm our proposed reaction mechanisms. And there are some experimental ways that you can confirm whether a proposed reaction mechanism is accurate or not. For example, in many of these proposed reaction mechanisms, we have intermediates. We described in the previous section what an intermediate is. So if we can identify the presence of an intermediate, for example, by spectroscopy or by gas chromatography, or by other methods, we can, for example, prove, okay, so there is that intermediate, and therefore that lends some kind of validation to our proposed mechanism. Another experimental confirmation is to look for predicted effect of conditions. For example, in some reactions, we propose that there is a step that involves the breaking down of molecular iodine, I2, into individual iodine atoms. Well, we know that this process can also happen simply when we irradiate iodine with visible light. So, if we carry out the reaction and the mechanism is correct, then we would expect that irradiating the reaction mixture with increasing intensity of light should speed up the reaction, because we know that the breaking down of iodine is affected by the presence of visible light, and we are proposing that that step is one of the key steps in our mechanism. Another classic method of confirming reaction mechanisms is the using of labeled reactants. What I mean by that is that you have manufactured a reactant that has a specific radioactive isotope at a certain position on the molecule. And we will study more about radioisotopes in the next unit. But for example, there are isotopes of carbon and of phosphorus that can be detected by different methods. And for example, starting with a molecule of glucose that has a C14 radioisotope at a specific position in the molecule, we can track what happens to that particular carbon throughout the metabolic pathway. Let's consider now the energy diagram for one of these sets of reactions that we are calling a reaction mechanism. As we can see in this diagram, we have a reaction that proceeds in two steps. Notice we start with the reactants on the left, and they have to overcome an energy barrier, an energy hill. That's what we call the activation energy. At the top of that hill is what we call the transition state, or the activated complex. We have proposed that this represents a state in which the bonds in the reactants are starting to break, the bonds in the products are starting to form, and so you're kind of right in between the reactants and the products. Notice that in this case, though, the products you get are not the final products of the reaction. There is like a small valley there before you hit a second activation energy peak, and that is the activation energy for step number 2. So notice that in between those two peaks, that valley, that is where we find the reaction intermediate. So to make sure we don't get them mixed up, the activated complex or transition state is a species that we propose that exists, but unless we have some very sophisticated methods for viewing what is happening to the molecules, we don't really see this activated complex. We don't observe it. However, the reaction intermediate is a real species. It is one that we can observe, we can even isolate, and therefore that is the big difference between a reaction intermediate and the activated complex. When we discuss the factors that affect the rate of a chemical reaction, one of the factors we mentioned then were catalysts. We briefly defined them, but we want to study them in a little more detail now. Catalysts are substances that affect the rate of a reaction without being consumed. They work by providing an alternative mechanism for the reaction in a way that has a lower activation energy. The way we identify a catalyst in a reaction mechanism is the following. Catalysts are consumed in an early mechanism step and then regenerated in a later step. So on the bottom left, I'm showing you a chemical reaction that involves the formation of oxygen molecules from ozone, O3, and oxygen atoms, O. This is a very, very slow process. However, in the presence of a catalyst, we see a two-step mechanism that makes the reaction faster. By the way, I've circled a symbol here, a double arrow that you may or may not have seen before. This represents a reversible reaction. It's a reaction that can go in both ways. Maybe it starts from left to right, and after a little while, when those products begin to accumulate, then the reaction starts going also in reverse until you arrive at something that's called a chemical equilibrium, and we will study that in a few weeks. Notice that in the first step of this mechanism, there is this monatomic chlorine, and notice that in the second step, that same species is released as a product. In other words, monatomic chlorine is consumed in the first step of the mechanism and then regenerated in the second step. And that is how we identify that this chlorine atom is a catalyst for this reaction. Let's see how catalysts work from the point of view of energy. A catalyst acts by lowering the activation energy. If you remember Arrhenius's equation, K equals A times e to the power negative E sub A divided by RT, where K is the rate constant, A is the frequency factor that addresses the fraction of molecules that have the correct orientation on collision, and E sub A is the activation energy, R is the gas constant, and T is the absolute temperature. We looked at this mathematically earlier and said that if the value of the activation energy decreases because this is present in this exponent of E, that results in K increasing. In other words, the reaction being faster. So let's look at the energy profile of what we call an uncatalyzed reaction. So here's again that energy diagram. A and B are the reactants, C and D are the products, and in between their energy levels there is a peak or a hill that is what we call the activation energy, E sub A. Now what happens if we run the reaction in a catalyzed form, in other words, in the presence of a catalyst? As you can see from the energy diagram, in this case, the activation energy hill is a lot lower. In other words, E prime sub A, the activation energy of the catalyzed reaction, is considerably smaller. So the result is that the rate of the catalyzed reaction is much faster than the rate of the uncatalyzed reaction because the activation energy of the catalyzed reaction, E prime sub A, is lower than the activation energy, E sub A, of the uncatalyzed reaction. Let's look at an example so we can see with numbers how this works. Let's do this practice here. I'm sorry this might not have made it into your handout, but please follow along and write it down if you want to. So we have a reaction at 25 degrees Celsius that has an activation energy, E sub A, equal to 335 kilojoules per mole. When a catalyst is added, the reaction has an activation energy, we're going to call it E prime, and it's going to be equal to 163 kilojoules per mole. How much faster is the catalyzed reaction? So we're going to start by writing out the Arrhenius equation for each one of these. Let's start with the catalyzed reaction. We're going to call the rate constant for this Kcat, and it's going to be equal to A. Remember the frequency factor that allows us to evaluate what is the proportion, what is the fraction of molecules that are colliding with the proper orientation. So A times e to the exponent minus E prime over RT, where E prime sub A is the activation energy for the catalyzed reaction. For the uncatalyzed reaction, we're going to write the Arrhenius equation straight up with the activation energy being simply E sub A. Everything else pretty much the same. When we ask the question, how much faster is the catalyzed reaction? Essentially, we're trying to figure out, so how many times faster? What is the proportion? So let's do the proportion of Kcat over K. And let's write out the corresponding expressions for E. So for Kcat, of course, is A times E to the power minus E prime over RT. And for K, it's A times E to the power minus E over RT, where E prime are the corresponding activation energies. We're going to assume that the factor A is constant, so we can cancel it out. And the rest of the expression, you should remember from algebra, right? E to the m divided by E to the n would be E to the m minus n. So when we subtract these two, we have a common factor, which is 1 over RT. And then you have minus E prime minus the negative of EA. So you should be able to work out to obtain the following expression. E to the exponent, E sub A minus E prime sub A, all of that divided by RT. Now, when they gave us the activation energies, notice that they were given in kilojoules per mole, right? But the constant R in its thermodynamic version has a value that is given in units of joules per mole per Kelvin. And so we're going to have to convert the difference of the activation energies into joules per mole. In the same way, the temperature here is given as 25 degrees Celsius. But in order for us to plug that value here, we had to convert it to the absolute temperature. In other words, the temperature in Kelvin. So once we put these numbers in and we carry out the operations, we're going to get the following expression. And again, feel free to pause the video and go back and make sure you understand where these numbers came from. All right? So we have the difference between the two activation energies, but we're converting that to joules from there. And then we have the value of R, and then the temperature, which is 298 Kelvin. Again, if you feel like you need to pause the video, go back, make sure that you understand where these values came from. And now, let's put these numbers in the calculator. It might be a little tricky, but what you get is the value 1.41 times 10 to the 30th. In other words, by dropping the activation energy roughly to about half of what it is for the uncatalyzed reaction, the catalyzed reaction can proceed at a rate that is 1.41 times 10 to the 30 times faster. That is an incredible change. That is an incredible increase in the rate of the reaction by simply dropping the activation energy to 1/2 of the uncatalyzed reaction. So when we study catalysts, we can distinguish between different categories. For example, in heterogeneous catalysis, the reactants and the catalysts are in different phases. And here are some examples of some processes. For example, the synthesis of ammonia, the production of nitric acid. and catalytic converters in cars, where the catalyst might be, for example, a solid, and the reactants might be, for example, gases. In homogeneous catalysis, the reactants and the catalysts are dispersed in a single phase, usually liquid. So many chemical reactions are catalyzed by the presence of acid or base. We will study these maybe later in the semester. The Haber process for the production of ammonia allows us to get around a lot of difficult thermodynamic issues in this reaction. The catalyst in this case is a solid interface made of several inorganic components. You can see them there listed. Typically, we list the catalyst kind of either above or below the reaction arrow. And as you can see, the catalyst essentially provides a surface that makes it easier for the molecules of nitrogen and hydrogen to break down or dissociate into single atoms, and this allows for them to combine and form ammonia, and that is the so-called Haber process. We're perhaps more familiar with the functioning of catalytic converters in our cars. As you know, if you have a combustion engine, the combustion doesn't always produce carbon dioxide as the end product. Sometimes incomplete combustion produces carbon monoxide that we would like to get rid of. The reaction would involve carbon monoxide and perhaps any hydrocarbons in the fuel that did not get burned properly, and finishing the combustion all the way down to the final products, which should be carbon dioxide and water. Now, what the catalytic converter will do is, when you put it in there, it will cause this reaction to go a lot faster. And again, the catalytic converter can be made of different materials. In the past, a lot of the material used was platinum, which made it a very highly coveted target for thieves. They'd steal cars and steal your catalytic converters so they can extract the platinum in it. Also, because the combustion engine works in open air, and the majority of the gas that's in air is actually nitrogen, you can also get some incomplete reactions with nitrogen that produce nitrogen monoxide and nitrogen dioxide, which are pollutants. And so the catalytic converter also helps to convert those back to just nitrogen gas and oxygen gas. Now, in our cells, there are many chemical reactions that happen in order to sustain the life of cells. And many of these chemical reactions are helped by catalysts called enzymes. Enzymes are typically large molecules, mostly made of protein, and they have a specific shape. Now, let's remember a little bit from your biology class what an enzyme is and what a protein is. Remember, a protein is what we call a macromolecule. In other words, these are molecules that are huge in size, from hundreds to thousands of grams per mole. And they are made up of subunits called amino acids that are linked together to form a chain. And that chain, because of the properties it has, it folds into a final three-dimensional structure that is the active form of the protein. So enzymes are a subclass of these proteins, and we're going to do a little cartoon here, and we're going to say that the enzyme is the cartoon there on the bottom. It has a particular shape. On its surface, there may be projections and crevices, and what we call here the substrate is the reactant in question. So in biochemistry, we call the reactant a substrate. And what you'll notice is that there are features on the surface of the enzyme that match the substrate, kind of the same way a key can go into a lock very specifically. That portion of the enzyme that matches the substrate and essentially binds it into this enzyme substrate complex is what we call the active site. For different reasons, the binding of the substrate to the enzyme causes the substrate to be more susceptible to the step that in the reaction that the enzyme is catalyzing, and therefore allows for products to be formed more quickly. The products are then released, and the enzyme is also released intact, ready to begin another cycle. The kinetics of enzymes follow certain patterns. One of the most common ones is the following. You start out with a first step in which the enzyme and substrate combine to form the enzyme substrate complex, and that proceeds with a certain rate constant. Notice that it is reversible, so in other words, it goes back and forth very rapid. However, the enzyme substrate complex is the one that yields the product in a second step of the mechanism, releasing the enzyme so that it can go on to another cycle. We could describe the rate of the reaction as a change in the concentration of product per unit of time, and the rate law typically is going to depend on the concentration of the enzyme substrate complex. In other words, that second step is typically the rate determining step, and so the rate depends on the concentration of that enzyme substrate complex. Now, I'm giving you a very simple model here. Many times that second step or the breakdown into the product might actually involve several steps all in there. If we look at the rate of formation versus the concentration of substrate itself, we find an interesting curve here. In other words, at higher concentrations of substrate, the rate of product formation increases, but it reaches a plateau. It reaches a maximum after which the curve flattens out. And the reason is at that point, the amount of enzyme available, right, is not enough, and therefore all of the active sites are occupied. And we say at that point that the system is saturated. Well, as you can imagine, since I told you that this is my field of biochemistry, I'd love to share with you some interesting aspects of these kinds of reactions. So, in many processes, it's interesting, but some of the enzymes that are used by the organism are called proteases. In other words, these are enzymes that break down other proteins. So, for example, when you consume a meal that has protein in it, let's say meat, for example, there are several proteases that are involved in digestion in the gastrointestinal or GI tract. The stomach produces an enzyme called pepsin, and then the pancreas produces other ones called trypsin and chymotrypsin. And here are three kinds of proteases. They're called serine proteases because at the active site is usually a amino acid called serine. And here is chymotrypsin, trypsin, and elastase. Now, you can see that at first view, even if you don't really understand what the diagrams mean, you know, the flat kind of arrows and the kind of like spiral kind of portions, don't worry about that. Just look at the overall picture and you can see that all three of them are extremely, extremely similar. All right, there's very little difference between them. Now, in a red color, you can see the portion of that protein or that enzyme that is the active site. Now, let me show you what happens. The active site of these different enzymes are different. Now, you could picture that it has a different type of cavity. And so, for example, in the enzyme chymotrypsin, you have an active site, in trypsin, a different active site, and in elastase, a different active site. In this diagram, what you're seeing in blue, black, and red, that represents not the enzyme, but the protein that's the target of the enzyme. And for example, let's say in the case of trypsin, the active site is such that it fits only the amino acid arginine, for example, and it fits in there, and that is what causes the enzyme to essentially splice or cut the target protein right where that amino acid arginine is. Notice that in the case of elastase, the target is a amino acid called alanine in the target protein. So it's a very interesting, very complex field. And you can see, for example, how it might work in this case. This is chymotrypsin. We're now represented in its overall globular shape. And then you have a protein chain, that's the target. And we're going to zoom into where the active site of the enzyme would be. And you can see at the active site, there is a portion of the target protein's chain going through there. Each one of those blocks represents a different amino acid in that chain. And once the enzyme arrives at a place that is recognized by the active site and binds the target protein, the enzyme changes shape, and that strains and weakens the bond. It's called a peptide bond between the amino acids in the target protein that exposes them to water for a reaction called hydrolysis, or breakage by water, to happen. The bond breaks, the enzyme releases the two halves of the protein chain, and then returns to its original shape. So that is a little cartoon of how an enzyme would work. So the study of enzymes is fascinating. I'm just giving you this as a little tour through them. But of course, you are not required to know all this stuff or memorize any of this stuff. Just remember that the way enzymes work is by folding into a particular three-dimensional shape in which there is a particular feature on its surface, a crevice, a projection, something that recognizes and binds to the reactant, what we call substrate, and allows the chemical reaction to proceed. That is the end of this lecture, and we have covered the topic of chemical kinetics. Please pay attention to the deadlines of the different assignments that are due, and also be on the look on Canvas for a practice exercise or a set of practice exercises that I am going to post a little bit later. Thank you so much, and now we're going to be ready for our next unit. We're going to talk about nuclear chemistry.